There. Hey, Professor Yost.
Yeah, what's up?
I just want to tell you I can't stay on here because I'm at the voting place and I've been here since 7:30 and I'm still online.
Good for you for going.
So yeah, I just wanted I think I put my video on, but the line's still pretty long, so I I had a reminder pop up on my phone, so I just want to tell you that's why I can't stay.
Been studying from.
No worries. Yeah,
There's-
I believe it.
You see that? Okay. Okay.
No worries.
Well-
Remember, we'll post a recording. So if you miss
Okay,
anything, you can always
thanks.
catch up.
Okay.
All
All right, thanks.
Good
Bye.
luck with that, Krista. Bye. All right, everybody. Let's get started. Sorry, I live in Bethany. Our power has been going out and coming back on and going out and coming back on. So I apologize if I get kicked off. I hopefully won't. It's been okay for a little while, so cross our fingers that it stays that way. Let me get this going. Let's see. All right. Alrighty. So we'll start with our week 11 review. Week 11 is dealing with chapters 17 and 18. We'll give our little brief run through of our topics that we've covered in 17 and 18, and then go over our most missed topics on those things. To be like, just to remind everybody, like 17, well, I guess we're going to do it right. So 17 is dealing with our immune pathologies. These are our immune diseases, our like hypersensitivities and hyposensitivities. Think of them as like your overreactions and your underreactions, right? So for chapter 17, a lot of like, especially when we're talking about our allergies with our type 1 hypersensitivities, we are considering the fact that the hygiene hypothesis has led us to this concept of essentially we've kept things so clean as a developed culture. We wash our hands so much. We keep, we shower every day. We cook all of our food to completion. We, don't, try not to, eat things out of the dirt. And we have shoes on for the most part when we go outside. All of these things, even like swimming in chlorinated pools and things like that, have it decreased our exposure to things like the parasites. So we don't see worms as much. We don't see the other parasites like the protozoa, like things like Giardia or things like malaria, even as much as we might have earlier on in the development of humanity or in undeveloped countries. So in those areas, they're getting exposed and having their immune systems trained on, hey, I'm a body, I can make IgE, I have eosinophils, and these things are used to attack, the parasites as opposed to attacking, pet dander or stupid things like that. So our immune system gets trained to deal with these things early on in our childhood. And as you become an adult, it's much, much more difficult to take back control of that, which is why the more you're exposed to as a child, like if you grow up on a farm, you're less likely, and you're going outside and you're playing outside and interacting with the environment, you're less likely to have allergies than somebody who didn't have as much outdoor exposure. And people who have grown up in these areas where they're having more parasitic infections like sub-Saharan Africa and in the tropics, these areas are more prone to parasitic infections, parasitic exposure, and therefore they are actually considerably less likely to have any allergies at all because their immune system is trained properly. So hygiene hypothesis, and the reason I'm even going on this much, I mean, of course, like if you guys get to the exam, you're probably going to have maybe one question on hygiene hypothesis, if I'm being honest, right? But to be aware of that and how the effect that our environment can have on developing basically how your body reacts to things, hygiene hypothesis is a great example on how your body will carry that development with it for the rest of your existence. So it's a big picture idea. All right, anyways, moving on to like our content on chapters 17 and 18. Chapter 17 had to deal with first, like we said, broken up into hyper and hypo. So hyper, we have our 4 types of hypersensitivity overreactivities. Type 1 was our allergies. Type 2 was our blood group stuff. So let's see, types of hyper. We had 1 was allergies. 2 was our blood groups. 3 was our immune complexes. And 4 was our delayed type. Excuse me. So allergy ones, these guys are dealing with an antibody, which was IgE, typically associated with our parasitic infections like our protozoa and our parasitic worms, our helminths, right? So normally that's used for that. Type 2 had to deal with our blood interactions. This is IgG and IgM. these antibodies being like produced naturally, because you have a type A blood type, you automatically make antibodies to type B. Your body sees those markers as foreign. It's never had that in their bloodstream before. So that's the idea with that. If you are type O, remember you see A and B as foreign. If you're type AB, you're lucky because you see A as normal, you see B as normal, and you see O as normal. O is our baseline for all of our blood types. So those antibodies being present in your bloodstream naturally, if you're AB, you won't have any of that. You will just be able to accept anything. So that also takes into consideration not just the A or B or O blood groups, but also whether you have the Rh factor. Rh is whether you're AB positive or AB negative. So that positive or negative is our Rh factor. And that actually has a much greater effect on the hemolytic disease of the newborn. So rejecting the baby because of the different blood type. That has a lot to do with that. All right, so the immune complexes, that's type 3, these are IgG and IgM as well. I don't know why I wrote them opposite order, but whatever, it doesn't matter. These are usually IgG and IgM. It can also be IgA for this one. IgA has like similar functionality to IgG, but it is only going to be found in the mucosa. So remember, that's going to be like your literal mucous membranes and things like that, where this is our secretory antibody associated with those kind of stickier, moist areas. So it is activated better in that environment than IgG might work. So it has a similar function. So not surprising that in those sticky, moist areas, this is where these guys might get caught up with the antigens that they bind to and create those immune complexes. Immune complexes, of course, are just going to be antibody antigen complexes that are stuck to one another, and they haven't gotten cleared out by the body yet because of their size. Typically, because they're like a middle size, they can get trapped in those really fine, tight capillary areas, like at the surfaces of skin, in joints, the fine capillaries that we might see delivering tissue to the heart itself, the fine capillaries that we see for toxin filtering in your kidneys, and all of that. And our example that we had was that acute post-streptococcal glomerulonephritis, where nephritis is the inflammation of the kidney, and that is caused by those immune complexes just literally getting stuck. And because antibody is like used like widely to signal for reactions of, remember what antibody does, it coats, it binds specifically with receptors to neutralize pathogens. It is part of opsonization, so it helps your phagocytes eat up the bad guys. It acts as an antitoxin by binding up toxins specifically. It can agglutinate, so it sticks to one, microbe or part of a microbe, and then another one at the same time, and creating all of these like big old clumps of microbes all stuck together. Or it can activate your complement pathway. And that's the classical pathway of activating complement, that protein system that exists in your body naturally, that once it's activated, creates a cascade of activations. Particle A causes particle B to be activated. Particle B causes particle C to be cleaved into C1 and C2, and then C1 and C2 cause these guys to be activated and these guys to be activated, and all this stuff down the cascade. And those proteins each have a function as well, whether that's in inflammation response, that'd be activating things like your neutrophils to come do more work, should create signaling pathways for those inflammatory cytokine signals and all sorts of stuff like that. Or the complement can also act as opsonization help to help with the phagocytosis. Or complement can, of course, the biggest issue that complement can do is the membrane attack complex where all those complement protein parts that you activated can come together and poke holes in the target pathogens, causing them to release all of their contents, and it will leak out of the cell and kill those or lyse those cells that are being targeted by that. Those are typically going to be like the bacteria that we're attacking that are covered in the antibodies and whatever. So that's complement. Just reviewing that because we got that test coming up next week. I want to be sure you're understanding like the role of antibody in that, and the fact that if your antibodies are binding on to antigen in your bloodstream and getting stuck in your capillaries, you're activating all of that complement pathway by that antibody being stuck there. So we've got complement there. We've got neutrophils that are getting activated by the presence of that stuff being stuck there and then releasing their signals, and all of this inflammation comes into the area, causes damage to the area. So we see redness in those areas. We see in that inflammation, we see cell damage because of those complexes, and the IgG, for example, sticking to its target antigen and getting stuck in your kidneys as your kidneys are trying to filter out the normal stuff, right? The waste. And so we have inflammation in your kidneys. It's acute because it is associated with you having had the infection. It can happen days after the infection, but it's usually associated with having that infection. And once you've cleared out your immune complexes, you should be able to recover from the damage done to your kidney, though it can be potentially serious enough if you already have weak kidneys, if you had a serious enough infection to cause permanent damage to the kidneys. But yes, that's after you've had a strep infection, antibodies against strep floating around, getting stuck with strep antigen in your kidneys when it's trying to filter out the waste. All right, so the fourth type of hypersensitivity was your delayed type. This is because it's delayed because it is cell mediated. So it depends on your T cell memory response. We know that when you activate a T cell, you make your effector cells that are going to be either your helpers or your cytotoxic ones that are killing infected or abnormal self cells. Or you can have your memory versions of those, or you can have your regulator versions that are going to help mute the immune response against self. Make sure you're not overreacting to self. And all depending on the signals that you're getting from the cells around you kind of drives how your T cell response is going to be controlled as well. Well, as it turns out, when you have your memory cells for your T cells, that's not enough cells to drive an entire T cell response when you see an antigen again. Your antibodies can get cranked out by your B cells, your memory B cells, right away. They can just start spitting out those antibodies while they may also be, increasing their numbers again. So it doesn't have a delay in antibody, but that's with B cell. That's the humoral response. With the T cells, they depend on the cell activity. The cells are the ones actually doing the things, not antibody proteins. So with your T cells, they're going to get activated and in their memories, right? And they have to re-proliferate again. They don't have to go through differentiation again. That part should already have been taken care of early on, but we do have to multiply back up our arsenal. So it's like we had all these guys that were really good at the fight in the original fight, and they have to re-recruit, recruits and train them to be just as good as they are. So it takes just a couple days, just a day to 3 days, somewhere between 1 to 3 days, as opposed to like the full week to a month to create the original response. So these guys are going to see like the oils from poison ivy stuck on the outside of your cells. It looks to them like they're being presented by your MHC1 screaming out, help, help, help me. I'm infected with the alien bursting out my chest. Please come kill me before the alien bursts out my chest. Raising their red flags. And it looks like that's what's happening on the surface of your cells with your MHC1 complex, and these cytotoxic T cells and macrophages will go out and attack at that site as though your cells are infected, when they're really just coated in oils and stuff from a plant. That's the whole entire reaction of poison ivy, is your body thinking that it's infected with stuff. And it's just how your immune system decided to interact with that oil or whatever. So once you start seeing that initially for like your second time you've ever seen poison ivy, then that takes a couple days for those cells to regrow. But that's when you're going to see your rash associated with poison ivy, that weeping, oozing. It's very clear that you've got cell damage going on with your delayed type hypersensitivities like poison ivy, because you have that oozing. That's your liquid being, released from your body that's supposed to be kept, inside of your body, releasing from that cell damage. We don't see that with your allergic response in the type 1, where you have hives. That's just itchy hives, but your integrity of your skin is maintained intact in the allergy response. So delayed type has everything to do with your T cell response and the presentation of it, and the pathogenesis of it. The damage is associated with those cytotoxic T cells destroying your self cells because it thinks that they are foreign. This is also how you reject any grafts that you would get from a transplant, because your helper, like your red flag molecule saying that you're infected, that red flag that you can raise to say you're infected and have the alien in your chest, that MHC1 complex, you're used to seeing that as MHC1 and seeing that as yourself MHC1. And that's part of your regulatory process for making sure you react to things correctly. However, if you get somebody else's kidney put inside of you, your CD8 toxic cells that attack infected self cells, they see the kidney from somebody else. Their red flag MHC1 molecule looks like it's holding up and something calling for help. It isn't, but because it looks slightly different, it looks like MHC holding up, help me, help me, help me, instead. So we start attacking that tissue as though it is got aliens in their chest. It's like if you watch one of those alien movies and you see somebody come out of the room with all of those eggs that with the face hugger things, they never got one on their face, but you might just assume that they did and take out the threat for that person. And that's basically what your cells would be doing to a graft from another person, just assuming that you are not the same as us. So we're just going to go ahead and kill you. And that can destroy tissue that's been grafted within hours, if you've seen that before. However, if you have never seen it before, you'll go through your initial latent phase of, activation and proliferation and differentiation. It'll take a while before you start actually rejecting that kidney, on top of the fact that you are on immune suppressants. So if you've never seen that before, it can take a long time. But if you've seen it before, it can be very, very fast. Just depends on that sensitization. And sensitization is essentially what most of these antibody and immune-related hypersensitivities are going to rely on. You have seen this foreign thing before. You took your time creating your antibody against this thing. And now the second time and every time that you see it thereafter, your immune response is faster against it. Just like when you get your vaccines, the idea is you promote that response and you basically negate that latent phase. You're introducing this thing to your body so that your cells can go through that latent phase before ever seeing the illness and create that memory response so that when your body sees it, again, memory is activated and you can make antibody immediately, which is why these 1, 2, and 3 hypersensitivities, type 1, 2, and 3, they are so fast. And type 4 is delayed because we have to regrow those cells. And they are cell mediated, not antibody mediated. So we basically covered everything for types 2, 3, and 4. Coming back to the allergy stuff, remember that allergies mediated by the IgE antibodies for the parasites require initial sensitization. You have to see the thing first. You have to get stung by a bee first. Make IgE against bee venom. And then the second time that you see the bee, your body will just have that fast response. So fast that if you guys have your MMR vaccines protect you so fast against measles. You can walk through the mall. You can go to the fair. You can stand in your voting line and still not get the measles because your IgG response is there being made immediately against the measles so fast that you never knew it even got in your body, could be in your body right now. And just from seeing the guy next to you and be, protected by your memory. However, it's so fast that when your body sees pet dander or sees dust mites, that it will create its antibody, the IgE memory antibody, so fast that you start having itchy, watery eyes, like right away, the itchy throat, or having an asthma attack or something like that, pretty much within minutes of being exposed to that, what your body saw as an antigen, right? So that's the downside to it. Your body wasn't trained properly to see those as not a threat. And so that can be triggered by 4 different categories of antigens: the inhalants, the contactants, things you contact, the injectants, things that are going to be put directly into your bloodstream. That's usually drugs that are injected, or like venoms that are introduced by, bee stings or bites from some certain snakes and stuff like that. Then you have ingestants that you're actually eating, like food allergies. We have atopic versus anaphylactic. Atopic is local responses that might include your skin or your respiratory system, for example, or it could be systemic. And here I'm talking about circulatory-based systemic. If it is circulatory-based systemic response, that's anaphylaxis. That can be deadly within minutes, and that is best treated with epinephrine to help decrease the bronchoconstriction so that we can actually breathe again, and decrease the likelihood of a shock response so you can get your EpiPen and decrease that reaction overall and save your life. We want to get that epinephrine in you fast, but remember, it's going to go away fast too, so you want to get right to a hospital to get better treatment. Antihistamines are great for decreasing that histamine release from those mast cells that are waving around those IgE flags, trying to interact with your allergen. Decrease the degranulation and release of histamine and how it's going to react with the tissues, creating that response. Antihistamines are good for that, but they take a while for them to get into your bloodstream, so they're not good for those anaphylactic all over systemic shock infections. Some of these types 2, 3, and 4. So type 1, we have to have IgE against an allergen. That's a very specifically triggered response line. But types 2, relying on IgG and IgM. Type 3, relying on IgG and IgM or IgA antibodies, just stuck with their antigens in your bloodstream, your tight spaces in your joints and stuff. Or type 4, with attacking self cells that look like they're infected when they're not. These guys are very closely related to the development of certain autoimmune diseases where we are attacking self cells when we shouldn't be. So attacking your myelin sheath, the cells that are creating the molecules that are creating the coating that protects your nerves. Therefore, attack those, cause inflammation of that protective coating, and leading to damage of the nerves. And that's going to be multiple sclerosis type of effect. And then, attacking the tissues in your thyroid for Graves' disease, attacking those, causing inflammation in those capillaries in the skin. You see the butterfly rash developing for lupus, attacking the tight areas in the joints and in other areas of the body, leading to overgrowth of bone as part of that inflammatory healing response, seeing that from rheumatoid arthritis. All of these are going to be associated with autoimmune responses for type 2, 3, and 4 hypersensitivities. And also, yes, the gut biome being important in training your bodies, what is a threat and what isn't a threat. So when you have major changes in your gut biome, you're no longer getting the correct signals for what is threat and what isn't threat, especially later in your life. So that's the hypersensitivities. The hyposensitivities, you have primary, so that's 1, and then secondary, which is the 2 degree. And primary, these are the stuff you have from birth. These are going to be mutations in your system that are going to make you more susceptible to infections from birth. Like you carry this in your genetics. Whereas secondary infections are going to be acquired somehow, either from infection or exposure to certain chemicals or certain cancers that can affect your immune response. So these are acquired somehow. My husband showed me a podcast. I'm not going to lie. And this is one of those moments where this is me on my pedestal. I'm not even going to try to hide it. Okay. But he showed me this podcast he saw on Instagram where it was someone who had HIV with undetectable levels of virus, and they felt like they shouldn't have to disclose the fact that they have undetectable levels of HIV to their sexual partners because they're undetectable. I don't know if you're aware of this, but undetectable means that the machines that are testing your blood cannot see that there is virus, which means that the tiny little vial of blood that you gave for the test didn't have a noticeable or detectable amount of virus in it. It does not mean that you don't have virus in your blood. It does not mean that you are not contagious. Undetectable just means that the machine didn't detect it. That's all it means. You can still be contagious. You should still be disclosing that to your partner. If you are HIV positive and you have undetectable levels of virus in your blood, I think you are a gross person if you do not disclose that to your sexual partners. You think that because you're undetectable, that you don't need to disclose that, you are gross. Just saying. That is not a personal opinion. I think that you are spreading a disease that you should be telling people about. If you are, especially when we talk about, you guys know, this is not the same thing as if you have a sexual partner and you have, diabetes, which you might have type 1 diabetes forever. You aren't going to pass that on to your sexual partner. You might have undetectable levels of HIV and still be able to pass that on to your sexual partner if nobody is acting protectively. Potentially, nobody's on PrEP medications, nobody's using protection, then you can still pass on HIV even if you're at undetectable levels, and that person will have HIV for the rest of their life. And if you didn't tell them that you had HIV, they just someday start getting sick from random infections, and they might not ever know that they were ever exposed to HIV. You're a bad person. Okay, so chapter 18. Now that we've got that out of the way, especially since a lot of you are going into healthcare industry, I would hope that you would advocate for being, it should not normalizing having infectious diseases and not telling other people about them when you can pass it on. Don't normalize that. Normalize it being acceptable to tell people about it. Normalize that instead. Okay, so chapter 18 is our tests. Off my pedestal. Chapter 18, that's our tests for phenotypic, immunologic, and genetic diagnostic tests, okay? So determining if your patient has the disease based on tests that tell you about expression of genes. So that's your phenotype. Remember, your genes might have you're carrying a gene for your A blood type from your mom, and you're carrying a gene for O blood type from your dad. But what your actual expressed blood type will be is A, because it trumps the O gene, right? So that's the expression. That's your phenotype, is having the A blood type. And you can be carrying all sorts of genes that you aren't expressing. And how does that apply to your diagnostic tests? Well, if your bacteria expresses this gene that allows them to make this molecule, this enzyme to break down certain antibiotics, then they are antibiotic resistant to ampicillin or something like that. That's their expression. And we can use those expressions of those enzymes or traits or whatever it is they're actually expressing and put them onto certain media or expose them to antibiotics or put them through these tests where we can visualize whether they're expressing this thing or not, usually by a color change or, a clearing area around your growth on your blood auger or your what is we're doing next, the starch plate or whatever it is, color changes on your mannitol salt auger or in your PR carbohydrate fermentation broth, like we all had in the lab. Chapter 18 relating absolutely directly to what we were doing in lab immediately following this week. So all of that stuff, our biochemical tests, our or we can also look at these as physiology tests. This is a Y. I don't know why I put an I, but there you go. Physiological tests, those things. We can also include things like Kirby-Bauer profiles. So your antibiotic susceptibility, what staph looks like on an antibiotic susceptibility plate for certain antibiotics versus what E. coli looks like on a susceptibility plate for antibiotics would look different. And that can be predictive of what organism you're dealing with for certain organisms, okay? And then phage typing. This is also phenotypic, looking to see whether or not a phage can infect your patient sample. If this bacteriophage, this virus that infects bacteria, only infects salmonella, and we show that it infects your patient sample is salmonella because bacteriophage viruses only infect one species of bacteria. So that can help you tell what bacteria you're dealing with there as well. Just like Kirby-Bauer, these guys are also going to be leaving like clearing areas where you've killed the bacteria. It's just we're doing that with antibiotic or we're doing that with bacteriophages that kill the bacteria. Also, we're just like with Kirby-Bauer, not going to do this very often with all types of species. This is usually only certain species that we can do this with, okay? But those are our phenotypic tests. Then we have our immunologic tests. So our immune-based tests all deal with the specificity of antigen interaction. And this is either going to be mediated by antibody antigen or the T cell mediated delayed type, right? So that would be T cell to antigen. It doesn't matter. Either way, this is immune response. So this is what's important. Immunologic or serological. So that's another term we can use for this. Interchangeable, okay, for our purposes. These are all going to be either direct or indirect. And direct is going to be, does the patient have the antigen? And indirect is going to be, does the patient have the antibody? We talked about this with ELISA, but this doesn't just apply to ELISA. It applies to most of our immunological tests, if not all of them, asking whether this patient has the antigen or has ever seen this foreign thing ever before. And we're measuring that by the presence of our antibody, which is a sense of memory of having seen that thing, as well as if you have recently seen something, you should have a lot of antibody against it in your blood. And if you have distantly seen something a long time ago, you might have antibody in your blood against it, but it's going to be at a much lower level. So now we can actually measure the level of your antibody in your bloodstream as a predictor of how recently you've seen that thing not just whether or not you have seen that thing. Measuring the concentration of that antibody in the bloodstream is called your titer, your antibody titer. But the tests that we can use for determining if you have ever seen something versus if you actually have the thing in your body might include things like immunochromatography, where we are looking for immune interactions and then color changes in a test. So this is going to be color change in the test based on antibodies we've generated against, let's say, HCG, which is our pregnancy hormone that pregnancy tests test for, human chorionic gonadotropin. If it is produced by your body, then you'll have that in your blood, in your urine, whatever, and we can pop that on a pregnancy test. And if your sample of urine crosses that line, there's antibodies there that'll cause a color change in the line if they see your HCG. If you don't have HCG, it'll just flow right over that line and not cause that color change. So pregnancy tests work that way, and home COVID tests work that way. Anytime that you have a color change on a strip like that, that's immunochromatography. Chroma is talking about color change. Then we also have talked about Western blot, which is using gel electrophoresis of proteins, moving them to a little membrane paper, and then looking for that specific band for HIV protein using antibodies and visualizing that. Very specific, very sensitive confirmatory test. ELISA is enzyme-linked immunosorbent assay, where we are using wells that are lined with antibody or antigen to detect if you have antigen or antibody, respectively, of course, back to our direct and indirect, and using a secondary antibody with an enzyme on it that's going to cleave a substrate and cause a color change if you're positive. And then we have the complement assay that relies on our classical pathway of complement that we talked about previously. That's your antibody-mediated complement pathway to test for basically the complement activity. And that's all you need to know about it, okay? And we have to, again, reprise that concept of specificity versus sensitivity, because we're talking about our diagnostic tests specifically. When you're talking about sensitivity, you want to be sure you pick up all of the positives that are possibly out there. So you're having a reduced number of false negatives. So you will not risk calling something negative when it's actually positive. You're going to be sure you catch all of those positives. No accidental negatives. If you're specific, you will make sure that the things that you're identifying are always positive and you're not calling something positive when it's actually negative. We want to avoid that as much as possible. So that's the difference between those. Remember that the tuberculin PPD skin test, where they inject a little bit of that tuberculin under your skin, that's a delayed type reaction for chapter 17, where your T cells may have seen tuberculosis before, their memory is reactivated, and within 24 to 72 hours, you'll have a swelling up of that spot if your T cells have memory of seeing tuberculosis. If your T cells have had memory of seeing tuberculosis, you've either had the vaccine or you've actually had tuberculosis. Those are the most common reasons for a positive PPD skin test. Of course, there is some cross-reactivity where some people might have a positive looking reaction and never seen it before. So that's why you might have to go get a chest x-ray to verify whether or not you've actually got tuberculosis latent and hiding out in your lungs, and confirm that. So that's why you might have to have that done afterwards. But it's all for the safety of patients that you may be interacting with and people that you could be potentially spreading that to. So we want to be safe. But that's an in vivo test done on living, tissue in your arm while you're alive. Our genetic tests that we had to be aware of all had to do with genetic sequences for identification purposes. So this is based on our known DNA sequences or sequencing whole genomes. You might not know what you're sequencing for, but it's so easy to do nowadays with how technology works. that you have an outbreak going on, that's a lot of people are getting sick, we can do whole genome sequencing on samples and determine a whole genome pretty quickly and give us also information about the genetics of that microbe at the same time for how we can best treat the infection. So that's useful. But most of our other genetic tests are going to depend on, does this DNA sequence match up with this pathogen or this cancer or whatever? So that's going to be utilizing PCR. Yes, if you have PCR, you have to know what you're looking for. Does this pathogen's genetic makeup line up with what I think it's going to be, E. coli, or does it line up with what I think it's going to be, this type of cancer? PCR, you still have to have a guess. You still have to know what you're looking for in order for it to be useful. You can't just say, let's just PCR it and see what it is. That's not a realistic mindset. So pop that out of your head and pop back in the idea that you have to know what you're looking for first. And so you'll use primers for specific genes in organisms to say, this person does have, in fact, what I thought it was, E. coli. Hybridization uses a similar line of thinking as our antibody testing. So instead of using an antibody to find if the patient has an antigen, we will be using this specific genetic sequence to see if that patient has that genetic sequence and matching it up, DNA to DNA matches by that complementary binding of the A to T and the C to G and vice versa. And those will be linked to sometimes a fluorescent molecule. You can do this in actual live tissue or with blood cultures and things like that. That's called fluorescent in situ hybridization, or FISH. Then we have our microarrays, where you can have literally an array on a microchip of DNA sequences, and wherever your patient sample is hybridizing, then that shows you that that patient has that mutation or it has that infection's particular DNA profile in that patient sample, and you can diagnose that way. Mass spec uses bombarding things with lasers to ionize your patient sample and literally break it down to the molecular makeup. This has this much carbon, this much hydrogen, this much oxygen, this much this, and then based on how much of each of those carbons, hydrogens, and oxygens, we can say that this person had glucose in their bloodstream. And that's what MALDI-TOF does. Then we have our imaging of our CT scans, our MRIs, our PET scans, and even x-rays to help diagnose things and avoid invasive procedures. So now we can just biopsy areas and then use those for our testing. Cool. So that's our topics that we covered, our summary. Let's move on to the most commonly missed topics within those. For chapter 17, commonly missed topic is going to be anaphylaxis versus atopic in our type 1 hypersensitivities. We went through these already. These are going to be IgE mediated. You've seen this thing before. You had your sensitizing dose, right? And that sensitizing dose created your memory B cells that make IgE. They can be re-triggered to make more IgE later on against that allergen, as well as you have mast cells in that area where you saw the allergen, your skin, your eyes, your lungs, whatever, that are just always holding on to that leftover IgE, just waiting to have it re-triggered again. That antibody is being held out like a sensor, so to speak, trying to detect any level of antigen out there in their environment. When they do detect it, they degranulate histamine and you have your histamine response.
Calendar. Effective study tactics.
Okay, I know.
Starts in 15 minutes.
Anyways, so sorry, my Alexa is telling me things because she always has something to say. So
15 minutes.
we have our sensitization phase of your IgE production, then creation of your mast cells with those sensors of IgE antibody that's normally soluble being held on the outside of those mast cells like a sensor device. Okay. They'll degranulate the next time that they see it again, really fast, immediately. The second they see it, they degranulate and release histamine, and you get that allergic response. When this is happening throughout your entire circulatory system. This leads to essentially a shock response. So you have decrease in your blood pressure. You'll have bronchoconstriction where you'll get less air into your lungs because your body's trying to protect itself. And this will lead to anaphylaxis. You might have hives developing on the skin, itching and swelling like you would see with a normal atopic response, a local response. But we have everything happening all over the body. You have your skin reactions, you have your respiratory, you might even have GI issues, and your cardiovascular drop in your blood pressure as well. So you can faint, pass out, have difficulty breathing, have hives, and yes, poop yourself all at the same time in an anaphylactic response. The best way to treat that will be with using epinephrine, like EpiPen shots. But the best way to avoid having this is to, of course, avoid the allergen altogether, or you can desensitize by introducing small amounts of that allergen to induce IgG response instead. So you can send signals. You can get injections with cat dander allergen, cat dander plus little chemicals that signal your body to make IgG instead. And remember, IgG being your workhorse, you're going to make more of it. It'll like basically absorb up all of those allergen molecules so that they never see the sensors held out by the mast cells. The mast cells just sit there holding out their sensory devices and never have to degranulate because they never see it because IgG took it up, and that's how desensitization will work. Okay. So sensitizing reactions act swiftly for allergy. Basically, you have to sensitize yourself first. And once you've been sensitized, you will have a very fast reaction in allergic responses because your mast cells are always there ready to degranulate. So which of the following is the most immediate and effective treatment for anaphylaxis, which is our severe type 1, our systemic severe type 1 hypersensitivity reaction? Do we treat anaphylaxis with antihistamines, oral antihistamines? No, that's going to take way too long to be active in your body. So we can't rely on that to keep you from dying, basically, from anaphylaxis. We need something faster. Intravenous corticosteroids, they're great for reducing inflammation that's already happened, but we want something faster that's going to reverse not just the inflammatory swelling of tissues and things like that, but also having other signals involved with reducing our, inflammatory response systemically as fast as possible. This is faster than oral, to be sure, but we still can't rely on that spread into the other tissues to help reduce the inflammatory response. What we want is epinephrine, which is signaling your body to immediately let me breathe more, immediately let me survive this moment. And then you can get to the hospital where they can give you IV corticosteroids and reduce your inflammatory response, right? Then you have time to deal with that. Our inhaled bronchodilators are only going to be targeting your lungs. And so we need to be sure we're not preventing any other vasovagal responses like fainting and things like that with our epinephrine. So it's just not enough. All right, so in the sensitization phase, this is sensitization phase. This is the first time you're seeing that allergen for our type 1 sensitivities. Okay, so when you're first seeing that allergen, what role does IgE play? Does IgE neutralize the antigen in the bloodstream? Okay, so IgE will bind to the allergen, but is it neutralizing it in the sense that it's preventing it from getting into where it normally would interact in your body? Or do they bind to allergen-specific receptors on those mast cells and basophils? We haven't been talking about basophils, but they do a similar action as what the mast cells do, okay? They can basically wear that IgE on their surface and use it as a detector for allergen. Do they activate complement proteins to destroy the allergen, or do they enhance phagocytosis of the allergen by the macrophages? So D, to be clear, D is describing something called opsonization. That's our term. I know it's a newish term to you guys, so I just want to bring it up. The complement proteins, that's our complement cascade. That's our classical pathway. Complement. It's not that, and it's not D. And then are we neutralizing that allergen as a, if we were looking at an antigen, right? You're looking at a virus and you want to neutralize that virus by binding on to COVID spike proteins, keep it from getting into your cells. That would be useful and great. That is not what's happening with our allergen. It is not useful and it is not great. Instead, what's happening is our IgE is sticking on those mast cells and basophils, which would be great if the IgE was trying to get histamine response to get rid of worms or parasites, but it's not. It's just an allergen. It's just a normal thing we shouldn't be attacking as a threat. So B is our answer. That's what's happening with our IgE. IgE is made and it binds to the surface of our mast cells or our basophils, and then they'll wait to use those as detectors of the allergen in the future. All right, so another topic that's sometimes missed is that concept of our hemolytic disease of the newborn. With hemolytic disease of the newborn, this whole, so here it calls it hemolytic disease of the newborn, HDN. This whole thing is mother attacks the fetus. This is because they're making antibody against the baby's blood. So here we have to have a situation where that mother's immune system will try to kill that baby's blood type. Blood type, just to be clear. All right, so this means that you have a problem if you are a mom and your blood type is negative of any kind, it doesn't matter, and the baby is a positive. If the mom is an A and the baby is a B, if the mom is a B and the baby is an A, or if the mom is an O and the baby is an A or B. Okay, so obviously here we're talking in the top about our RH groups. That's what an RH group is. And then these down here are going to be our ABO groups, which we talked about when we were talking about our type 2 hypersensitivities. It is the most severe reaction with the RH group. Can you have rejection of a baby because of ABO groups? Yes, but it is typically much less severe than with the RH group. So if your baby is RH positive and you are an RH negative blood type, then we will give you RhoGAM to prevent your antibody response against the baby. Because like we said, your body automatically makes antibodies against a foreign blood type. If you're Rh negative and you've never seen this plus marker before, you see that as foreign, you'll make antibodies against this. With your first pregnancy, that is a sensitization reaction. We make antibodies against the Rh plus group, and the baby initially there might not be any serious sort of effect on that first baby. We're just making our antibodies at first with the first baby. Okay. The antibodies can cross that placenta and attack the fetal red blood cells and destroy the blood cells of the baby after we have seen our initial sensitization. So typically we'll see that with the second baby. So this happens during pregnancy or during childbirth, but once we can cross that placental barrier, okay? This is going to be more likely if you've had issues in your pregnancy. If you've had any bleeding in your pregnancy that could compromise the, crossover of these, especially if we are talking about having a miscarriage where we've already introduced compromise of that placental barrier, then you are more likely in a future pregnancy to have this reaction than somebody that didn't have that sort of a trauma in pregnancy carrying. So you can have babies survive this, or the baby could die from this reaction. So usually we will have jaundice, anemia, maybe enlarged spleens on the babies that do survive, and then something that they call hydrops fetalis, which is where we'll have fluid buildup in the fetal tissues, and that can be life-threatening to the baby. So that is the idea of how hemolytic disease of the newborn is going to work. Again, it's typically whenever the mother has never seen this antigen before, and the baby has the antigen. So the mother's antibodies are made against that antigen, and the antibodies go and do their job against the antigen, which leads to complement lysis of that baby's blood cells, and eventually basically shock, systemic shock, and hemolysis within that baby that can be fatal. We prevent this with RhoGAM to block that antibody reaction against the baby's quote unquote antigens, right? And we can also monitor for ABO situations, just monitor that situation, use phototherapy for the bilirubin that's made during that jaundice reaction, and use, in very severe cases, maybe transfusion. So if you are a mother who is a type A blood type and you're giving birth to an AB baby, that baby has B antigens, your body might react to that B antigen if it's your second B baby, right? And that baby might be jaundiced. And you have a baby and your baby's jaundiced, and your doctor is like, oh, this baby's jaundiced. And that's about all you get as information. Well, it might be possible that you had rejected that baby's blood type during your carrying your fetus. So that is one explanation for why your child might have jaundice after childbirth. It's treatable. Put them in the little lights to help get rid of the bilirubin, and they get to go home and they don't care that your antibody ever attacked them. And you might not have never known. But if you're having an RH incompatibility, the risk is death of the fetus. It's much more severe when we don't want to approach that. So helpful rhinos always share pink babies. It's crazy sounding phrase, but helpful rhinos. So helpful being that hemolytic disease, rhino being that RH factor. Always, it can also affect our ABO blood types. And sharing the pink babies, we're literally talking about, our symptoms, our sensitization for sharing. Pink is going to be prevention, how we're going to prevent that in the babies that's affecting your babies. So that's your little mnemonic for that one. Our questions for this one: which of the following is the primary method to prevent hemolytic disease of the newborn due to RH incompatibility? So remember, with the RH groups here, we're saying mom is RH negative, baby is RH positive. And so this is a situation where the mom sees the baby as foreign, and the best way to do that is to block that antibody interaction with our RhoGAM. So we're looking for RhoGAM in our choices, and that's going to be B. Talked about RhoGAM, none of the others do. It's not going to help you to give blood transfusions. It's not going to help you for vitamin K to the newborn, which would help with that bilirubin situation, or using antiviral medications during the pregnancy has nothing to do with a virus. Not helpful at all. All right, so the next question: a newborn is diagnosed with severe jaundice, anemia, and an enlarged liver shortly after birth. So this is already symptoms that we were talking about with that hemolytic disease of the newborn. The mother is AB negative. Okay, so she has the universal ABO group. She's not going to see a blood type, any ABO blood type anyways, as foreign. So it's not the ABO that's going to be the problem here, but she would potentially see positive is foreign. So that's what we're looking for here. Baby's blood type being A positive. So that's the problem. So mom is AB negative, baby A positive. So what are we attacking in the baby? The RH group. So what is the most likely cause of these symptoms? Is it RH incompatibility leading to hemolytic disease? It's going to be that one. We just deduced that from the question. Is it ABO compatibility? No, because that mother is AB. So she's going to see A as normal, B as normal, and O as normal. So it's not that one. Is it viral hepatitis? That's not what we're talking about here. Or is it maternal diabetes? That's also not what we're talking about here. This is clearly a blood type issue. So it's going to be A. All right, chapter 17, our tissue graft rejection. This is autograft versus allograft versus xenograft versus isograft, right? So autograft, that's going to be whenever you have tissue from self, you have a burn and we get skin from your back to repair the burned tissue on your arm. That's an autograft. You're not going to reject that, but we can only take so much skin from your own body. So that can be a problem. But as far as rejection goes, you will not reject it. For an isograft, this is an identical twin. Also very, very low risk of rejection. It's almost as good as the self. There might be some very, very minimal risk of rejection, just because of how, people's immune systems respond to their environments. But the likelihood of rejection is super, super, super low. So identical twins are great. Isografts are great. Allografts are going to be from other people, not self and not identical twin, right? So there's a risk that you're going to see that as foreign and reject that graft. And then we have xenografts. This is going to be from non-human sources. So like heart valves from pigs or from cows, bovine tissues and whatever. We can also use animal skins as a mean to cover burns when you have severe burns over your body, and we cannot use your own skin for it. So there's a high risk of rejection because this is a possibility of seeing that as foreign tissue. And there's also a risk for zoonotic infections, so viruses that might be in those animal tissues. So which type of tissue graft is most likely to require lifelong immunosuppressive therapy to prevent rejection? So you're not going to need to suppress your immune. Don't pay attention to this. You're not going to need to suppress your immune system if you're getting a tissue graft from yourself. So it's not going to be A. Is it an isograft? You're probably not going to reject your twins, your identical twin stuff. It's very, very unlikely. So you probably don't need it for that. Now for allografts and for xenografts, typically your body will eventually see a xenograft as not a threat, just like it sees your gut biome as not a threat because it's just so different. It doesn't see MHC to attack MHC on that, right? There's no MHC1 on animal cells that we see as MHC1. That's a human thing. So you're more likely to have a lifelong issue with trying to get rid of the tissue if you have an allograft as opposed to a xenograft. So your issue is actually with getting other people's tissues as opposed to even animal tissues. Up front, you might reject that xenograft, but you don't have that MHC foreign issue of constantly thinking that something's calling for help with an alien in its chest. You don't have that with xenograft. You only really have that antibody response that can be regulated eventually over time. You're going to need to be on immunosuppressants for the rest of your life to prevent rejection if you are getting tissue from someone who is not self or identical. Okay, next question. A patient receives a heart valve transplant from a pig. Which type of tissue graft is this? And what is a major concern associated with it? So you get a heart valve from a pig, obviously, but we're just going to write it for our visual purposes, is not human. So that is a xenograft. And anything that is not human, xeno with that X is how we would describe that. And graft being a tissue transplant. And then what is a concern with that? We mentioned in our little mini review just a second ago, rejection is a concern or zoonosis. So these are animal diseases that can cross over to people, okay? All right, so autograft, is that an autograft? No, because that's a self to self. Is it isograft? No, because that is identical twin to self. Is it allograft? No, because that is different human to another human. And then xenograft is going to be animal to human. There's a high risk of rejection initially and a potential disease transmission risk as well. So it's going to be D. For chapter 18, for our diagnostics, our most missed topics. First one is going to be the different diagnostic test types, phenotypic versus immunologic versus genetic. Knowing what tests fall into what category is a commonly missed thing. So we're going to start with the immuno. This is going to be anything dealing with antigen interaction. So this could be antibody to antigen. Does the patient have the antibody? Does the patient have the antigen? And we're going to check that with the antibody. Or it could be T cell related with the antigen. It's usually going to be dealing with like your tuberculin skin test, right? So those are our immuno tests. It's always dealing with somehow interacting with antigen. All right, our genetic tests. These guys are always dealing with DNA sequence. So if we talk about PCR, what are we doing in PCR? We're amplifying DNA. That's a DNA sequence issue. If we are looking at DNA hybridization, we are using DNA probes to look for a DNA sequence. These are all DNA related things. A microarray is a microchip with DNA sequences on it to see if your patient sample matches any of those sequences. We can diagnose different cancers that way as well as infections that way, but they're all DNA sequence based, okay? So if your test depends on antibody and or antigen, it's immuno. If your test depends on the DNA sequence being tested, that is genetic. If your test is anything else, think of it as phenotypic. But our biggest examples, these are going to be your stains and your biochemical tests. This is going to be your antibiotics. So Kirby-Bauer. And it's going to be your phage typing. Those are the ones that we gave examples of in the lecture, right? So looking at things in a microscope, looking at your color changes in your media, looking at your Kirby-Bauer results, and looking to see if viruses infect bacteria. That's it. So does any of that have to do with actually testing DNA sequence? No. Does any of that have to do with actually testing your antibody or antigen interactions? No. Okay. So I don't know why, but people miss those a lot. You want to be aware of if it is not immunological, antigen related, if it is not DNA related, and it is phenotypic. Biochemical tests, color changes on your media, those are phenotypic tests. If you can't remember what phenotypic means, at the very least, remember what immuno deals with and what genetic deals with. If you can't remember what immuno deals with, I don't know why you're even in Unit 3, because that's all Unit 3 is immuno. We're dealing with your antigen interactions, seeing things as foreign and trying to get rid of those foreign things. So keep that in mind, please. All right, example questions. Which type of diagnostic test would be most appropriate for identifying a bacterial pathogen based on its genetic material? We're looking for DNA sequence here. Does Gram staining look for DNA sequence? No. Does ELISA look for DNA sequence? ELISA is enzyme-linked immunosorbent assay. Well, guess what kind of test that is? It's not DNA. It's immuno. PCR, does that look for DNA? Sure enough, it does, okay? Then we have biochemical test strips, like those ones that are going to change color, just like your media changes colors. That is not looking for your DNA sequence. That is not your answer. Genetic is always looking at DNA sequence. seems obvious, just actually apply that obvious literal definition to your test types and you'll be good. A patient undergoes a test to determine the presence of antibodies against HIV. What type of a diagnostic test is being used? Anytime you have antibody to look at antigen, which is exactly what's going on here, we want to know if the patient has the antibody. So we already know this is even further on an indirect immunotest. And we're going to use known antigen to do that. This is an immunological test. We're not looking at the genetics. We're not looking at the DNA sequence. We are not doing a biochemical test for presence of certain enzymes causing color changes. And this is not a phenotypic test, which, by the way, biochemical tests are phenotypic tests, right? So it's not going to be that either. So it's our immunological. Finally, we have our sensitivity versus specificity. Sensitivity, we are going to be seeing all the positives so that we don't have any false negatives. And specificity, we are detecting only the positives. So you'll have no false positives. So we're only looking for true positives and calling them positive. And we're not going to say something's positive when it's not. Okay. Sensitive snakes see small numbers safely is just trying to remind you that in sensitivity, you'll get even the smallest amount of things. Whereas if you were looking at specificity, we're not looking at the small amounts of things. We're only looking at the exact trueness, the truth of them. A diagnostic test has a sensitivity of 95%. What does this mean? All right, so sensitivity. We're going to decrease our false negatives. And we're going to hope to increase catching all the positives. Okay, so what does it mean when you are 95% sensitive? I would hope that that would mean, what I'm going to infer to this is meaning is that whatever that test is doing, it's going to identify 95% of, if there are people who are sick, we will catch that 95% of those people who are sick will get called positive. Okay, that's what I'm assuming that this means. Because sensitivity, it isn't always going to be 100%. The goal is to catch all of your positives, even the absolute weakest ones, but even the really, 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 really weak ones. You're pregnant for 4 hours. You go take a pee test, it's going to pop negative. That is a false negative. But they say those things are 99.9%, accurate. That means 99.9% of people who are pregnant will test positive. But sometimes you'll get tested negative in that 0.1% because it wasn't sensitive enough. So that's what that is talking about. So is the test correctly identifying 95% of patients without the disease? No. We're trying to correctly identify patients that have the disease with our sensitivity. Does the test correctly identify 95% of patients that have the disease? This should be it. 95% of the people that have the disease, they all, like 100% of the people have the disease. The test sees that 95% of those people have it. So 5% are not getting caught. Those are our false negatives. That's what we're talking about. So the test has a 95% chance of being positive in healthy individuals. No. Or the test has a 95% chance of correctly diagnosing the disease when it is absent. That doesn't make sense. Right? So that's basically like saying that these, people that don't have the disease, there's a 95% chance that we're going to catch that they don't have the disease. That doesn't make sense. So we're really looking for correctly identifying that 95% of the people that have the disease have it. We accidentally missed that 5% of them didn't have it. That's our false negative rate. Okay, that's what this is talking about. So a new test for tuberculosis is designed to have high specificity. What is the primary benefit of this characteristic? Remember, specific. No, that's a C. Deals with making sure that you're getting the true positives only and decreasing false positives. Okay, so for our TB test, the test will detect a high percentage of people with TB. That's sensitivity. So it shouldn't be that one. The test will accurately confirm that a person does not have TB if the test result is negative. Okay, so we're in the right direction here. So we want to be sure that our negatives are negative and our positives are positives, right? We don't want to be calling somebody negative if they are positive. That's more of a, I would say, more of a sensitivity thing. We also don't want to be calling somebody positive if they are negative, right? The test will have a high rate of false negatives. It shouldn't have effect on the false negatives. Here we're trying to decrease false positives. The test will identify a high number of non-TB infections. This is a strange way of kind of approaching this, but that's probably not going to be it. So we're going to mark C and D out. So that's essentially D is saying that the test will identify a high number of non-TB infections, saying that the test says, hey, this isn't TB, it's something else. That's not what the test is doing. That's a weird approach, but whatever. So back to B, the test will accurately confirm that a person doesn't have TB, and we're right about that when they're actually negative. That's the idea with specific. Somebody's negative, we want to be sure we're calling them actually negative and not accidentally calling them positive, which would be a false positive. So it's going to be B. I know it's a lot. So that's the end of the review for the most missed topics for chapter 17 and 18. I'm going to bring this up really quick to see that if I got any questions, because it does not pop up, and I apologize for that. It doesn't pop up with my chat during my lectures on this. So yeah, I'm so sorry that you're having to deal with all of this. Tammy, I hope you feel better soon. Truly, truly, truly. Please take care of yourself. I'm so glad that you're able to join us at least a little bit today. I really hate that you're having to go through that. It's the worst. All the good wishes to you and your recovery, hopefully soon, really. Let's see. Okay, what am I trying to do here? Immunology stuff. That's right. So we're going to go through the immune system really quick, hopefully really quick. I'm going to try to keep it as brief as possible because I don't want to be on here anymore either. So briefly, a run through of the immune response. Okay. And the idea is I'm going to try to, let me lock my screen in place because I know I'm going to turn this in weird directions. I'm going to try to go through from innate through into our adaptive response. All right. So first things first, let me get the thinnest line I can possibly get. We're going to start in our upper corner, upper left corner with our innate response. Your innate response, remember, you're going to have 3 defenses in your immune system. Your first line of defense might be things like your skin. This is really the waterproofing protein of keratin, chemical defenses like lysozyme, sorry, I have hiccups, or acid in your stomach. Things like this, right? These are all your first line of defense. They exist there. They help keep the microbes out. They might also be doing other things. Next, we have our second line of defense, which is your actual innate response. Okay, so you are walking around out in the fair, at the fair, and somebody has the flu near you and they're coughing. And um at the fair, that virus gets into your body, right? So you inhale as your first step in your infection process, inhale your virus, inhale the flu. All right, great. So normally your mucus will trap it in your lungs, and maybe you'll cough it out and get rid of it as well. Right, that should be happening. That's your first line of defense. That could be helping get rid of it. However, the flu virus has what we call a virulence factor on its surface called neuraminidase. And that neuraminidase is going to degrade down your mucus and make it more liquidy and make it easier to infect cells. It's also going to break down certain proteins on the surface of cells to help it get in as well. So it's going to basically like subterfuge this whole mechanism. Okay. So it's going to get around that because of its virulence factors. Remember, a virulence factor, and you guys should be giving virulence factors in your personal presentations. A virulence factor is anything that helps aid that infectious process and cause disease, okay? So virulence factors of this virus are going to help it into the body. So it gets into your self cells. So this is what we would call infection, right? All right, so if this virus, while it is infecting your cells, is multiplying, one of the things that it might be releasing is its double-stranded RNA genome, which is something that double-stranded RNA is something that our innate cells see as foreign immediately. Now, we don't have any double-stranded RNA in our own cells, so that's why this can happen. We have single-stranded RNA as our messenger RNA. Really, the hiccups are getting to me, and I apologize, but our single-stranded RNA as our messenger RNA, or our weirdly folded like transfer RNA, and all those things, we're used to seeing that. But actual double-stranded, like helix type RNA would be weird. So whatever this virus is, it has a double-stranded RNA genome, and our innate cells don't need to see that as an antigen. They just see that generally for any virus that has that, has nothing to do with it being the flu, nothing to do with it being a specific antigen. They can just see that general aspect that you have double-stranded RNA as foreign, right? And that is called a PAMP, which is a pathogen associated molecular pattern. PAMP. Great. And these PAMPs can be recognized by any of the immune cells that are like just hanging out in the area. These could be any number of for example, our granulocytes. And our granulocytes these could be any of our fills, including our heavy hitters, which are our neutrophils, or it could be our mast cells. which these guys, what they really do is send out signals, right? They degranulate. Or release cytokines, which are our cell-to-cell signals. Great. So they release those cytokines after seeing the PAMP, and that calls in our neutrophils from the bloodstream. So our neutrophils squeeze through between our blood vessel cells and move into that area, being called in via chemotaxis, so we're drawn there by chemical signals, right? We squeeze through those blood vessel cells into the tissue with our fluid. That mechanism is called diapedesis. All right. I don't know, whatever, phone. Okay, so they are now getting called to this area by those chemical signals. They move into that area via diapedesis, and they're moving there with fluid from the bloodstream and other components of that fluid, the liquid. This is like more like I'm talking about like fibrinogen that's going to help with clotting, other like chemokine signals that are helping signal the whole process. It could be complement that is there as well, but whatever. The other things that are in the blood besides our neutrophil and our other cells. So we're going to move into that area with the fluid into those damaged tissues. Is called exudate. And then build up of exudate leads to like a firmness in that area, right? Firmness and swelling. And that is what we call edema. So already with this swelling in that area from that exudate building up, this is what we call tumor. That's our word in our inflammatory aspect, right? Also, whenever our mast cells are degranulating and sending these cell signals with these cytokines, one of the other things that they might be signaling for is vasodilation or constriction. And the reason you might want to constrict is to keep the pathogen from leaving that area until we get proper help in there to help eat them up and break them down and whatever. But these signals for vasodilation can lead to redness in that area, that increased blood flow and warmth. And also increasing the fluid as well in that area for the same reason that you have fluid moving in with the neutrophils. So the redness is called rubor, and the warmth is called calor. And the damage in the area itself can lead to pain, which gets some little kind of a little offshoot here, pain, which is called dolor. So these guys here are 4 Latin words associated with inflammation. And remember, the inflammatory response itself has a purpose. It's associated with calling the right cells there to the area. And it helps with bringing our healing, basically, well, I don't want to refer to it, but like calling for the healing molecules and the healing cells to come in to repair. So we'll say the repair process calls for defense and starts the repair process. Excuse me. All right. So that's what inflammation is for. And it also keeps you aware that you have something going on. So you're a larger organism. We've got cells working together. Now you know that something's going wrong in your body because you have pain and redness and swelling and warmth there. Okay, great. So we've got our inflammation process going on while we're calling in our neutrophils, which are heavy hitters as far as our phagocytes go. So that brings us to phagocytosis. And essentially, we got called there first by that chemotaxis. Next, we're going to recognize the PAMP. And that's going to lead to ingestion. So that's that process of eating the literal phagocytosis portion. Now we're going to create a vesicle where we're going to contain it. And that is called a phagosome. And we're going to merge that with a lysosome that contains toxic stuff. And that's going to create something called a phagolysosome. And that's going to kill that pathogen. And then we're going to excrete our waste from all of that. So we're going to come back to the presenting of the waste because neutrophils don't do that. So right now, our neutrophils are just going to excrete the waste. So the presenting thing is really only going to be done by our macrophages, which are our crossover cells, macrophages and dendritic cells. They are innate cells that signal our adaptive response. But our neutrophils are only going to be excreting, okay? So we went through, we're a neutrophil, we came in there, we were called in there by our chemotaxis with our chemokines, our cytokines, same thing. And we are now going through phagocytosis of the viruses that are outside of the cells. We already recognized our double-stranded RNA that was released from the cells whenever we burst out, or from parts that our infected cells are like releasing or presenting, or whatever it is. Our neutrophils are seeing double-stranded RNA and saying, hey, this looks like a foreign thing. And now we're going to start eating up all of the foreign material in this area that we recognize with this PAMP. And we're going to eat it up, break it down and kill it, and excrete the waste. So that's what's happening in there. Meanwhile, we are still releasing, while we're doing that, still releasing cytokines to call for more help. Plus, we are also excreting our waste, which is also going to be looked at as antigen. Okay. All right. So next, we have some things that can happen. Let's follow that antigen, shall we? Antigen moves out of tissue so that lung area where you inhaled your flu virus, it's going to move out of that actual lung tissue and into the lymph. Remember, we have an injured area, so the fluid that's coming out of that injured, inflamed area is going to move into the lymphatic system. So now we're going to go to a lymph node. And it's like our checkpoint. And it's at the hangar where our B cells are going to be. All right. So here's your B cell. And it is, we'll call it naive. It's never been activated. It is just waiting for signals, right? Cool. And on its little tiny surface, it has a receptor called IgD. Remember, Ig means immunoglobulin. Great. So this guy can bind onto, well, it's waiting in that hangar, waiting at the checkpoint. And while everything from the lymph is getting made sure it can pass back into the bloodstream, this naive B cell is checking everybody's passport. And one passport that it sees is that antigen. Okay. So it sees that with its little IgD receptor. Its receptor sees exactly that antigen. Now, notice that I've called this an antigen. It is not a PAMP to this. To this, it is an antigen. It is a very specific antigen to, it will be eventually antibody interaction. Okay. So it recognizes that part of the antigen. And that thing is called an epitope. Remember, that's the part of the fidget spinner that we're actually binding with on our fidget spinner antigen. So we've recognized the part of the flu spike protein called the epitope. So now our naive B cell has seen the antigen that it needs to be activated. But is that going to activate it? No. All right, so this is essentially the end of our innate response. Okay. There will be some crossover and some cross reactivity, of course. But essentially, that's the end of our innate response. All right. So now we're going to move on to our adaptive. And now I'm actually going to start from the bottom and go up because we're at where our B cells are here at the bottom. So let's just work up from there. So from number 5, we're going to go on to number 6. Here we have our naive B. They had IgD with this antigen. And what it's going to do is take this on into the inside of the cell and then move it out to the surface of the cell. Or this blue guy here. Because we found the antigen hanging out on the outside, was the B cell infected with the flu virus? No, right? It picked up that antigen because it was waste made by the neutrophil that was left outside, flowed through the lymph. B cell picked it up from outside. So it's going to present it on a molecule called MHC. I'm going to use the number 2 here, okay? But know also that it could be the Roman numeral. Okay. But I don't want you to get confused when I start writing it all out because my handwriting is terrible. So it's going to present it on MHC2. Well, doing what? To what? To whom? What's the purpose of that? There's another cell that can come into contact with it, and this is our T helper, or TH, right? And it is also naive. It has never seen this thing before, this antigen. It has never interacted with it before. It's just been waiting its whole little tiny life to engage with this. So this has something called a T cell receptor. It's a lot like the antibody thing, but it is always going to stay attached. So this is our T cell receptor. And it's going to recognize that antigen. That antigen that's being presented on that MHC2, MHC2 is a general molecule that presents any antigen. It is not antigen specific, okay? Our T cell receptor recognizes a specific antigen. All right, so here comes our naive T helper cell that's going to recognize that antigen specifically. Okay, so it binds onto that. And when that happens, it has a molecule that's going to make sure that it is seeing MHC2 while this is happening. And this molecule is called CD4. CD4 recognizes that that is MHC2. It knows that this is the correct response that's happening. The T helper cells receptor says, yes, this is antigen that I also recognize. You and I are on the same wavelength. We are interacting with the same antigen. You're presenting it the right way. So now this T helper cell can activate, finally, that naive B cell. So it won't be naive anymore. And that B cell can simultaneously activate that naive T helper cell. So this is great. So we're both stimulating and activating at the same time. That's the benefit of having your B cell available to present antigen on MHC2. Now remember, MHC2, let's actually keep with this. It has a couple of rules. It's only going to be found on antigen presenting cells. And these are our B cells, our macrophages, our dendritic cells. And if you want to be specific, these guys here are also known as derivatives of monocytes. Which are a type of agranulocytes. In fact, all of these guys here are antigen presenting cells, are agranulocytes. Another agranulocyte would be the T cell, because B and T cells are lymphocytes, and lymphocytes are agranulocytes. So lymphocytes and monocytes in their derivatives are agranulocytes, okay? All right. I'm going to put a little note about this also being the T cells are not APCs, but they are granulocytes. So let me go ahead and do this so that we don't get confused. Okay. Okay, so we've got this going on. Let's keep with on our story, okay? So number 7, your B cell, once it has been activated, will go through proliferation to make several B cells. B. These are all just B cells that have proliferated. The next thing that these guys are going to do is undergo, all of this is called differentiation. So these will become B memory, B regulatory, or plasma, which are our effector ones. And our plasma cells are going to make soluble antibodies. And of course, we're making our antibodies. Well, I'm not drawing this particularly well, but so this is antibody. And these can be three different types. The first one is going to be IgM always. And then it will switch to either IgG or IgA or IgE. We already introduced IgD, right? That was our receptor one down here in step 5 and 6. So now we have all 5 of our antibodies. Our first one is always IgM. IgG is your workhorse. IgA is your mucosa. And IgE is your parasite and allergy. Okay. So that's what's going to be going on with your B cells. Great. This, your naive T helper cell, it does stuff too in step number 7. Okay. It also makes T, that's I, T, H, T, H, T, H, right? So this will be your proliferation response for these guys. And then this guy here might become a memory one. This guy here might become a regulatory one. This one is a little bit trickier though, okay? And this again is our differentiation. This is an A step as well, okay? So this will become our effector. So that's our one that actually does the job for that cell. But T helpers have 2 different effectors. They have TH2 and TH1. Okay. TH2 guys are typically involved in helping the B cell response. Keep in mind, you already activated your initial naive B cell and it turned into memory regulator and plasma. Congratulations, okay? I don't know what else you want me to say, but that your immune response is always adapting. You have still other B cells that recognize other epitopes. You still want your IgM to turn into IgG or IgA or IgE. All these signals are going to be helped by that TH2 response. So it doesn't only activate B cells and not just one B cell. It'll help activate other B cells and help stimulate that antibody production in the direction that we want it to go. So that's what TH2 is going to do. TH1 has a whole other job, okay? And we're going to come to that. Our step 9 here, and again, I'm arbitrarily numbering these, okay? Break it down how you like it, but I'm just trying to keep it orderly. Step 9 is now you have a self cell that is infected. In your lung cells, you had cells that were actually physically infected. When they got infected, they might have been releasing virus out that was budding out of the surface and whatever else, getting released out into the system, recognized, broken down by your neutrophils, pooped out by those, ran into the lymph system, saw the B cell, and now we've got this activation happening. But you also still have cells that are being infected, like in real life. So your infected self cell that has the antigen in it will on its surface be presenting on this molecule here, which is your MHC1 molecule. This stuff comes from the inside. And still, we're presenting our antigen. I keep doing this, I'm not meaning to. Okay, so we're presenting this on MHC1. This is our red flag. And this cell is literally being like, help me, I'm infected. That's what that MHC1 is saying, okay? So outside of all of this stuff that we just saw down here with our B cells and our T helper cells and all of this, we have now a cytotoxic T cell that is naive. On its surface, it has, this should look familiar. This is its T cell receptor that will recognize that antigen. Okay, so here he comes, and he's going to come and recognize this antigen. It sees that and says, yep, I definitely recognize that. Cool. He also has a molecule on his surface. It looks like this. And this molecule is called CD8. And CD8 will recognize MHC1. The T cell receptor recognizes the antigen. And when we have this going on, does this activate the T cell? It does not. Okay, so step 10, what we're going to have is signals from our T helper 1 that was activated by our antigen presenting cell on the MHC2 that picked up the outside stuff. Remember, this is full circle now. Bad guy came in, Innate saw that double-stranded RNA. We activated our neutrophils. Neutrophils ate it up. The crap left over circulated into our lymph system. Our B cell is our antigen presenting cell. It picked it up on its little IgD immunoglobulin surface receptor, saw that antigen, presented it out on its MHC2 because it came from outside, the MHC2 interacted with the naive T helper cell, which then activated both the B and the T helper. T helper differentiated into its types, including Th2 to stimulate our B cell response, and Th1, which is now sending signals to activate our cytotoxic T cell response. Our T helper cells only ever see MHC2. They interact with MHC2 via their CD4 receptors, which is the only type of molecule, CD molecule, that the T helper cells are going to have that we're interested in. Okay. CD4 recognizes MHC2 only, and Th1s and Th2s have CD4. But now they're activated, they can create cytokines to stimulate their other responses. The cytokines the Th2s make help with the B cell response. The cytokines the Th1s make help activate your cytotoxic T cells. So now in step 10, let me move it up a little higher. Now in step 10, if I take a copy of this, I really don't want to have to redraw this, so bear with me for a second. We'll just get all of this, that's fine. Actually, do it though. Okay, well, this is not annoying at all. Okay. Trying to copy it. Okay, let's see. Okay, there we go. That's what I'm trying to do. Okay. Get this up here. All right. All right, so now we've got this stuff up here, and now we actually can activate this guy with our Th1 response. So what is going to happen is our Th1 gives the signal to activate this guy so that he will proliferate to TC, 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 and then these guys are going to differentiate to TC regulators, TC memories, and TC effectors. Right? So that's the idea of what is happening once we go through this activation here. We're going through all of this, right? So we've gone through that proliferation and we've gone through that differentiation. So now our actual effector cytotoxic T cells, which can now come and interact appropriately, they can now they will make, I don't want this, stop that, stop, solid, good, undo, make granzymes and perforins. That's what these dashed lines are, okay? All right, and that perforins will poke holes and granzymes will degrade the insides of our self cells so that they will go through apoptosis. Which is cell suicide. Okay. Just being real here. So they will decide that they don't need to be alive anymore because they have gone through so much damage. So that is essentially the overview of the entire immune response. This makes way more sense watching the video of this and me drawing it out actively as opposed to trying to figure it out looking at this, because this is just going to look like words and sketch. So if you are looking at this at the end of the video, please take the time to go through from the beginning. That was really good handwriting. But that's our adaptive immune response versus our innate immune response. And how we got from one to the other. And in the middle of all this, we're also signaling back and forth all the time with one another, telling each other to help stimulate our inflammatory response or help to stimulate the B cell response or help to stimulate our cytotoxic T cell response, whatever it is, for making antibody against that antigen or killing infected cells or whatever that is. So that is our overall summary of our entire innate and our entire adaptive immune response for chapters 15 and 16. So don't forget, we still have chapters 11 and 12 for our control and our antibiotics, and we still have 17 for the pathology and 18 for the diagnostics, and that's all of Unit 3. So I hope that this was at least somewhat helpful. Of course, I'm going to post the video. I'll post this slide as well, even though it's a mess, but hopefully that makes some kind of sense to you guys whenever you are studying with that too. So having said that, let me check because I know I got chats and again, I apologize to you guys. Yes. So Melina asks for sensitivities. Would it be best to paraphrase a bit due to how much information there is in regards to presentation? Absolutely. When you guys are presenting your stuff on your whatever your topic is, whether you're going over your chapter 17, for example, so that's what that is. We have our hypersensitivities for type 1, 2, 3, and 4. If you guys want to paraphrase those and say, have your slides, recommend that you have your slides can have as much information as you want on the slides. So you can say you checked all your boxes for covering everything. The absolute barest minimum that I want everybody to be covering in your content. So if you are a team that is using PowerPoint slides for your presentation, what I want to see is if you've divided up, this person has type 1, this 2, this 3, and this person 4. Then if you're covering type 1 hypersensitivities, that you cover all of the bullet point information on the actual you are welcome, Jen, on the actual study sheet at the bare minimum, and then include any information you need to help explain the bullet points that were on the study sheet on your slide. And then when you're actually presenting, summarize that, and at least the slide has any of the additional information that can help explain the details that you're summarizing. So most details on your slide. Your explanation helps bridge the information on the slide with the basic bullet points on the study sheet, if that makes sense. So you definitely want to cover that stuff on the study sheet since you're meant to be like a sort of review person on that topic. So that's the goal with that. Yeah. So you'll always check those boxes if it's on your slides. If you're explaining the summary and I see that your summary makes sense in your mind coming out of your mouth, I'm probably not going to ask you questions about it. If you're just reading your slides, we might have questions, right? So we know how that goes now. We've seen it. So keep that in mind for your presentations. So I'll get all of this posted as soon as I can for you guys. Thank you for bearing with me. I will see you again on Sunday we'll do the gameplay review for that. So this is, that's mostly just going to be gameplay. I do have a surprise for you guys. That'll be 4 p.m. on Sunday, like we usually have been doing for the other units. So 4 p.m. on Sunday via Zoom. I do have a announcement about that that's on there. I'll probably send another one just so that you guys are reminded again, because I know things happen between now and then. So yes, I think that's everything. But yeah, I do have a surprise coming definitely by Sunday. I will have the surprise made available for you guys. It's not candy or something, okay? It's a study tool, but yeah. And I know we had said for our group presentations that we would try to have, like, I couldn't get the university. I tried to contact the, yeah, I tried to contact the marketing department, see if they could come out and do the videos and stuff, but they're on other projects right now. They didn't have anybody available like during a class time that I had. So we won't have somebody videoing, but I will have some little mini gifts, we'll say little tiny things that you're probably going to throw away, but hey, it's fun to get the trophies and whatever. And we'll take pictures of the groups that we vote as having our best presenters for Unit 3 that will be put on the wall in the micro lab. As you know, so for each section, you'll have a winner, if you will, for the best presenters for that. So we'll have something there for you guys as your little mini trophy winning, you did the best of everybody for that unit. So keep looking forward to that too. It's a little thing, but it helps. So yeah, so I'll get this review posted. I'll get that slide posted. Sunday we'll do our review and I will get your little study surprise out. And then I will see you guys on Monday and Tuesday for our presentations. Wednesday and Thursday are the exams. You should have gotten an email from me telling you that if you are taking the exam in the testing center, that I have put your stuff in the testing center. If you want to take your test in the testing center, then, and you have not gotten that email from me, please email me ASAP so that I can get you scheduled for the testing center. I'll say this again in class. Hopefully I'll remember to say it again in class on Monday and Tuesday as well, because that'll be like our last chance, other than people with like issues with computers. If you take your exam in the test in the SB 100, like we have class in SB 100, that lecture hall, SB 100 has terrible internet reception. So our exams for SB 100 will be in SB 101, and I will re-announce that on Monday and Tuesday as well. But I don't want you guys to be surprised when we have to be in that other room. The internet is better in there for whatever unspeakable reason. I have no idea. So that should be everything. Huh. All right. I think that is all. So we got, yes. Cool. All right. I think that's everything. Any other questions from you guys?
No more. Thank you very much. Lots of infos, but we'll try to absorb it more.
It is a lot. Yes.
Thank you.
Yes. Thanks, Jen. All right. All right, guys. So I'll see you guys on Sunday. Have a good weekend. Get some rest.
Thank you.
Bye.